Thank you. I'm really grateful for the opportunity to present to you all. Really, as Livia says, I've been following a lot of your work for some time, and maybe we can talk about that a bit in the discussion and how it relates. But I'm focusing now on what I've been doing, sort of in the last 5 years or so, trying to kind of understand placebo effects across the spectrum of psychiatric disorders. Um, So just to say, I am supported by the National Institute of Health and Social Care Research in the UK, but otherwise I don't have any other conflicts of interest that are relevant. So I tend to start my talks slightly differently in that I tend to spend quite a bit of time justifying why looking at the placebo effect matters. And I'm going to do that slightly differently in this talk for this audience because I think I don't need to spend a lot of time talking about how powerful these effects are. Um, but I am going to talk a bit from the angle of treatment discovery in psychiatry. Um, and this slide is from the WHO's Global Burden of Disease studies. Uh, and I'm just putting this here to make the point really that as we've kind of as a health system dealt with communicable diseases, infectious diseases uh, as a kind of major burden of disease, we're now finding that the major contributors to health burden are things like psychiatric and neurological disorders, particularly depressive and anxiety disorders, rank very highly in the kind of burden of disease. The We have treatments for these disorders, but they're far from ideal. Many patients don't respond to first-line treatments. So something like 50% of patients with depression and anxiety will not respond, or they experience unwanted side effects. The trouble is, we're not doing very well at developing new treatments to kind of address those problems. Um, I had a look at FDA approvals for new drugs over the last 10-ish years, and kind of looked at some different fields. And if you look, oncology are doing pretty well. They've had 140 new treatments approved in the last 10 years. Um, Neurology, which is a kind of brain adjacent or adjacent to psychiatry anyway, specialty. 48 new treatments, which is not bad. Um, and anyone want to hazard a guess as to how many we've had in psychiatry? Novel drug treatments? If you don't want to hazard a guess, just think about the answer. 16. Oh, there was someone in the chat just put something in. What did they say?
Zero.
Zero. Wow, zero is a bit pessimistic. 16 we've had in the last 10 years, which is not many. And if you spread that out across the various disorders we've got, you're kind of looking at one or two treatments per disorder. In fact, I know for anxiety disorders, there has been zero. Um, so, uh, so we're not doing very well. And if you look at the average time from kind of compound discovery to use in the clinic, psychiatry has that longest average time of about 10 to 15 years. I think it's about 12 years, um, which, and that's an average. So there's some that are much longer. Um, and there's a few different reasons for why the treatment discovery is kind of so slow in psychiatry. Uh, One, I think we still have a real lack of understanding of the neurobiology of the disorders so that we can really kind of target mechanisms in psychiatric disorder. I think there's a topic for a whole other presentation at some point around the outcome measures that are used in psychiatry and whether they're really fit for purpose. And I think that is a major problem. Uh, and the other is the large placebo response that we see in clinical trials in psychiatry. Um, and it is large and clinically relevant. Uh, this is from a meta-analysis looking at anxiety disorders. And you can see the effect size of placebo response across various anxiety disorders in RCTs over time. It is large, effect size is mostly above 0.5, and is increasing over time. You see a similar picture in schizophrenia. So this is antipsychotic trials. On the left, you can see placebo arms. On the right, drug arms. Drug arm response is fairly flat, but placebo response is increased. And the problem you've got there is then your delta, so your kind of signal that you're detecting in your trial between your drug response and your placebo response is getting smaller. And so detecting any efficacy gets harder. So I recently asked the question, how large actually is the placebo effect in psychiatric disorders? And is it the same across disorders, or is it different? We carried out an umbrella review. So this is a review of reviews looking at placebo effect size in psychiatric disorders. So we searched for systematic reviews and meta-analyses that reported an effect size within group for placebo arms in RCTs. We actually identified quite a few papers that were potentially eligible, but we were worried about potentially double counting data. And so for that reason, we excluded meta-analyses that did not have the largest sample size or the largest number of trials. So that if we only included the ones with the largest number of trials, you tend not to duplicate data. So although we found 50 meta-analyses in total, we excluded 30 that didn't have the most information. So we looked at 20 meta-analyses across psychiatry. And I should stress that's not comprehensive. That didn't cover every psychiatric condition. So there's more work to do out there. But this was what we found. This is a summary kind of forest plot looking at effect size of placebo across different disorders. So you can see the disorder listed here, subpopulations if they were reported, and then effect size. There's quite a range. So the generalized anxiety disorder came out with the largest effect size, 1.85, which is very big. All the way down to OCD, where you're kind of looking at 0.3. I think the important thing to say about all those effect sizes, though, is that they are all, in the clinic, they're all clinically relevant effect sizes. They would be clinically relevant changes on symptom scales and so on. So all of these placebo effects are relevant. We also looked at predictors that were reported across different meta-analyses. I couldn't fit all this on a slide in an ideal way, and I've put this here just to highlight actually how variable across disorders it is. A few things do come out in multiple disorders. Number of sites, for example, comes out in a couple of disorders, possibly because of some variation that occurs as a result of different recruitment practices and so on. There was some sample size as well, came out in a couple of places, but also look, publication year comes out as a positive predictor in panic disorder, schizophrenia, anxiety disorders, as we mentioned before, alcohol use disorder. So there's something going on as well in relation to that. Just to kind of sum up that point, really, and I don't think I need to explain to this audience, but the placebo response is made up of really multifactorial things. Partly kind of statistical artifacts, partly placebo-specific mechanisms. And what we really don't know still, and what I'm trying to understand is how much of the placebo response that we see in clinical trials in psychiatry is made up of each of these different parts. So to bring me to what I'm broadly going to talk about, I'm going to talk about how I've been trying to understand expectation effects in trials for anxiety disorders and how that relates to blinding. Um, I'm going to talk about what we know about neurobiological mechanisms, particularly in anxiety and depression, how placebo responses happen. Um, and finally, I'm going to talk about how I attempted to develop an experimental paradigm to look at placebo anxiolysis to try and understand it further. Um, and I'll let you know how that went. But to start with, kind of looking at expectations in anxiety disorder trials. We know expectations matter in psychiatric trials, but we only know it indirectly because it's hardly measured. But you can kind of see these effects. So here's an example. On the left is a meta-analysis looking at antipsychotic trials. And what was quite interesting about this analysis was they compared placebo-controlled trials with low-dose controlled trials. So that is using a lower dose of medication as your control with active controlled trials. So that is no placebo arm. The control was a well-known antipsychotic medication. And what you need to focus on really is the dark grey bars. So that is patients that received an effective dose of medication. And you can see that the response is lower in placebo-controlled trials than it is in active controlled trials. And there's kind of a dose response relationship depending on the dose of the control. And I think the inference you can make there is that as the belief that you will receive an active treatment is more certain, if you're in an active controlled trial, you only get active treatments, then your improvement is bigger. So it suggests that expectations do play a kind of causal role in response. Likewise, you see a similar thing in depression. So this is a graph showing response rates on the y-axis, probability of receiving placebo on the x-axis. And you can see the response to both placebo and antidepressant is lower when the probability of receiving placebo is higher compared with when the probability of receiving placebo is lower. So as the certainty of the treatment goes up, it seems the response also goes up to a point, because then there's a bit of a drop off at this 20% or less. Although that's kind of indirect, there have been kind of direct tests of the role of expectations. This is one example, kind of using a variation of the open hidden paradigm, which I think was quite ingenious. This was by Faria and colleagues in 2017. They recruited patients with social anxiety disorder and essentially gave them all a well-known treatment for that, which is escitalopram, which is an antidepressant. And they essentially randomized people to a different explanation about that treatment. So half the patients were told, this is escitalopram, good evidence-based treatment for social anxiety disorder, and it should improve your symptoms. The other group were told, this is an active placebo. So you might get side effects, but it's nothing to do with it being a drug. That's purely to mimic some of the side effects of the real drug. And what you can see is that the group that were told they were receiving escitalopram had a much bigger response compared with the group who were told that it was an active placebo. In fact, the effect size between the two there is 1.2, which is a very large effect, much bigger than you see in most clinical trials for social anxiety disorder. So that just shows how big the effect of kind of expectations can be. But obviously that's not the situation you tend to get in clinical trials. You tend not to get one group where expectations are boosted and another group where they're actively reduced. So can we see this effect of kind of open hidden kind of effects within clinical trials? Well, there's some evidence that maybe you can, in that not all placebos seem to be the same, or in the same show the same kind of efficacy anyway. You can see this across anxiety disorders. So again, this was from the Bandelow and colleagues meta-analysis. looking at different treatments for anxiety disorders. Um, and what was quite interesting was they compared the different control groups as well. So you can see pill placebo here outperformed a psychological control, which outperformed waitlist. Um, and possibly the reason for that is because it's pretty hard to blind waitlist. Um, it's pretty obvious if you're on the waitlist. In the same kind of way, blinding psychological treatments can be quite tricky as well. So maybe there's a kind of unblinding effect going on that may explain some of these different effects. We also see this in depression, and this has become quite a big debate in depression because of kind of psychedelic treatments being investigated. Some of you may be aware of this. So psychedelics cause kind of hallucinogenic trips, and there's good kind of serotonergic reasons for why they cause those trips. And so there's a whole kind of a field of research exploring whether these can be used for certainly as antidepressants, but possibly for other conditions as well. This was from a Bayesian meta-analysis exploring placebo-controlled psychedelic trials with placebo-controlled escitalopram trials. And what's quite interesting is depending on what the comparator is, the placebo performs differently. So here the comparator is placebo response in antidepressant trials. And what you can see is the psychedelic placebo uh, performs worse with an effect size of about 0.2, um, which is a not insignificant effect size. And possibly the reason being that it's quite hard to blind if you're on the placebo psychedelic and you don't trip, that's quite obvious. Um, but if you compare placebo antidepressant with extremely low dose psilocybin, they're actually almost identical. So, and the extremely low dose psilocybin, it causes some effects that may be noticeable. In fact, some patients do report quite a big effect on that. So maybe it's a better blinded control. So potentially unblinding effects do matter in kind of clinical trials, and that might be because of expectations. Broadly, there's two kind of types of unblinding that can happen in a clinical trial. So one is kind of benign unblinding, and this is the idea that if someone gets better because of a treatment, then they might unblind themselves as the fact that they're on the treatment. And that's kind of okay because the unblinding is directly related to the efficacy of the treatment. In which case, if that is measured in your clinical trial as an effect, then you can say, well, that's probably causal and related to the treatment effect, and that would be okay. However, you can also have malicious unblinding. This is the idea that if you improve or if you don't improve, and you attribute that to either being on the treatment or on the placebo, so you experience a side effect, for example, that causes you problems, or you don't experience a side effect, which says to you you're not on the treatment, that might change the way that you rate your symptoms or that somebody else rates your symptoms, which can then lead to artifacts and problems in your analysis. How do you know that the treatment effect is true and is not just because of malicious unblinding? So I got quite interested in, because of all this that was going on and lots of debates about this. I got quite interested in trying to understand whether unblinding actually does affect efficacy estimates in psychiatry, whether this is benign or malicious. Um, and I think the short answer is we don't know whether it does or not. But some attempts have been made to explore this. So there has been analyses, meta-analyses looking at whether blinding has been assessed in clinical trials for depression, for example, and whether that's associated with outcome. In fact, it's been done twice. Um, there's also been reviews looking in ADHD, which I actually helped with, and we only found one study that looked at blinding integrity in ADHD. So I decided to also look at this in anxiety disorders. So, can we see whether unblinding in trials of anxiety disorders affects the outcome? So to do that, we tried to collect every single RCT that has been done in an anxiety disorder since 1980, which we were reasonably successful in doing. We managed to get 248 trials, um, and we had to look through all of those and see whether any of them reported any kind of blinding assessment. Because what I was interested in was the difference, kind of the effect of the blinding on the outcome. Um, and only six had any kind of report. So out of 248 trials, six published a report of blinding integrity. Um, we then, with a student of mine who was very determined, contacted every author that we could on all those to try and ask them, do you have any blinding data? And actually, three authors got back to us and said, yes, actually, we do have some blinding data. So we managed to get three more. So in total, we had nine RCTs reporting any kind of blinding assessment, which is not many. Um, so what we then did was took those assessments of blinding and calculated a blinding index. So we, because the assessments we had only had correct or incorrect guesses, there weren't any don't knows, actually made calculating the index quite simple. So we took the number of correct guesses minus the number of incorrect guesses over total guesses. And when you do that, you have scores closer to 0 suggest intact blinding, scores closer to 1 suggest perfect guessing, scores closer to -1 suggest opposite guessing, which can also suggest unblinding. And so we could then use that in further analyses. And we calculated this index for clinicians and for patients separately because we basically hypothesized that actually the unblinding happens differently for clinicians and patients. So we calculated the index for each group and for different raters where we could. This was what we managed to do. So two of the studies, there wasn't actually enough information to get Bang's blinding index. So they reported things like kappa rating or what have you. But that did end up in the narrative synthesis if you want to find the paper. But in terms of what we could actually collect and calculate a blinding index for, this was what we found. And I think you can generally say the clinicians know whether the patients are on medication or placebo. Particularly these studies. These studies actually were in MAOIs, and I think the side effect profile is very obvious. So that may explain the perfect guessing that went on there. Among the patients, again, they're much better at guessing when they're on the medication arm than chance as a general rule. But the placebo arms were much better blinded on the whole, which was kind of encouraging. So we then entered this into a regression. So I was interested in, does the effect size, does the Bang's blinding index, influence the effect size that you get, the difference between medication and placebo? So we looked at this for clinicians first. On the y-axis is effect size, standard mean difference. On the x-axis is Bang's blinding index. And you can see there was a positive relationship where the more unblinded the trial, the bigger the effect size between medication and placebo. So negative numbers here favor medication. So again, this doesn't necessarily tell us whether there's been malicious or benign unblinding, because it may be that, people have guessed that the patients are on medication because they've improved, or because it could be the side effect profile of particularly those two treatments. In the patients, we get a slightly different story. So we found in the medication arms, there wasn't really a relationship between unblinding and effect size, but there was, at a trend level, there was a relationship in the placebo arm, whereby if people were unblinded in the placebo, there was a bigger difference between medication and placebo. that could be malicious unblinding. Um, so I think Luke Gellin has coined this term, which I quite like, and he's a clinical lecturer. I think he's a clinical lecturer now in King's, um, general adult psychiatrist as well. He said this idea of kind of nocebo, or it's sometimes called placebo. You kind of know you're on the placebo, and that disappoints you because you're not getting the treatment, and that may worsen your symptoms. Um, and it's possible we've seen a signal there that that's happening in at least in anxiety trials. Um, so I've written a bit more about this in a narrative review that published earlier this year in JAMA Psychiatry, which you can look at if you're interested. But essentially, what I said in there were these kind of recommendations, um, which was that we have very limited data. In fact, we have no data about expectations and limited data about blinding integrity in psychiatric trials. But there's signals that suggest malicious unblinding might be a problem, um, which is an issue that we need to think about. Um, I think we should be measuring expectations at baseline, at least. And I say at least because I think expectations are likely to change as the trial goes on. So I think we should be measuring that routinely. I think we should also be measuring routinely whether blinding was intact and what the reasons were for failure, because without that, it's very hard to know whether if unblinding has happened, whether that's for benign reasons or more malicious reasons. So I think we need to be having that so that we can better interpret the kind of clinical trial results that we get. Um, so that's kind of me trying to understand how expectations might be influencing clinical trials, and I think it's fair to say that without measuring expectations, that's quite tricky, and I think we do need to do that. In terms of biological mechanisms, what biological mechanisms might be influencing placebo responses or placebo effects in patients with psychiatric disorders, and I think we have quite a good understanding of some of the biological mechanisms in kind of placebo analgesia. Certainly, we know endogenous opioids are involved, possibly dopamine. Um, and I was interested to know what do we know about that in psychiatric disorders? And I focused on depression and anxiety because the placebo effects are quite large in those disorders. We tried to do a review. The idea of it originally was to do a meta-analysis, but we couldn't do that in the end. But we tried to review all the data to see where it's reported a change in imaging in placebo arms in patients with anxiety and depression. Uh, can we see any correlates or that I signify any kind of mechanisms that might be relevant? Because those could be useful targets for treatments, right? Um, or could be useful for kind of exploring in the lab. Um, so I was interested in trying to find that out. There's very little data. We identified only 15 studies, 12 in depression, 3 in anxiety disorders. Um, there's more papers, but they're actually repeated analyses of the same kind of population. So it's hardly explored, is the best way to put it. Most of these studies as well were post hoc analyses and very correlational in nature. They didn't look at causation. So, does this mechanism affect the outcome? It was more this person improved, what changed in their brain. But the signals that do come out are that ventral striatum and dopamine seem to be important. In fact, the evidence for this was probably stronger than the evidence that came out for the opioid system. But there were also signals that might be involved as well. And so I guess the question is, why is there so little data when the placebo effects seem to be so large in these populations? And it could be that there's a lack of experimental paradigms, right? If it's hard to kind of induce these responses in people in experimental settings, then it can be quite hard to image. Um, And there is work going on in this area to try and improve that. Winfried Reef and others have been looking at placebo mechanisms in depression through kind of acute inductions of sadness, which is quite a nice way of doing it. Um, and I was interested in seeing whether we could develop like a similar kind of paradigm, but for people with anxiety disorders. And there has been one attempt to do this before. Um, this was by Benjamin Mayer and colleagues, developed this kind of threat of shock paradigm with a kind of repeated placebo that was involved. It's quite a complex paradigm. So there were these kind of threat trials with the red squares where you had a chance of being shocked with an electric shock. And there were these no threat trials, which were the green squares, and then kind of alternating lorazepam nasal spray that was actually saline versus a normal saline spray, um, looking at kind of threat responsiveness. Um, and there were some effects that were maybe kind of a placebo effect. The main issue I have with this paradigm is that if you're going to be shocked by something, that's quite a an adaptive. If you get anxious about that, that's quite an adaptive anxiety response, quite a rational one. Whereas the patients I see with anxiety disorders, their anxiety is completely irrational. It doesn't make any sense, and they know it doesn't make sense, but they can't stop it in some ways. Um, so I was kind of thinking, really, if we wanted to model the clinical picture that we see, we need a paradigm where we can model that uncontrollable, uncontrollable anxiety. It just so happens that in Southampton we do have a model where we can model that uncontrollable anxiety. Um, and what we do is we ask participants, healthy volunteers, to come into the lab and inhale air that's been enriched with extra carbon dioxide. So we increase the concentration of carbon dioxide up to about 7.5%. Oxygen content is the same, so people don't suffocate, but it does seem to cause a kind of generalized anxiety picture. And this has been replicated many times. Breathing the gas both increases kind of subjective anxiety, but also physiological measures like blood pressure and heart rate. And you can also see psychological changes in threat processing as well. And it responds very nicely to standard treatments. So if we give people an anti-anxiety drug like lorazepam or an SSRI, we see that their anxiety response is less in the model. So it seems like quite a good clinical model. And I wanted to see if we could use this to condition placebo responses. Um, this was the way I went about it. So I brought in 32 healthy volunteers and gave them a baseline carbon dioxide inhalation so that they could get used to that feeling. And then they were randomized to one of two groups. So in one group, they were told that they were going to receive a lorazepam nasal spray, which is an anti-anxiety drug. In reality, that nasal spray was contained only normal saline, so salt water. We chose a nasal spray because we thought we could give the story that it would act quickly, that the nose is connected directly to the brain. Would also explain why kind of we thought that it would only last sort of 20 minutes or so, which is about the length of an inhalation. So we gave them that spray, and then we said we're going to run the inhalation again. But when we ran it the second time, we replaced the carbon dioxide with normal air without their knowledge. So they were just breathing normal room air. And then afterwards, we gave them another dose of the placebo nasal spray and said we were going to repeat the inhalation again because we were interested in repeated dosing effects. And this time, we repeated the inhalation with the full carbon dioxide. So we were interested in, does the change in anxiety from here to here show a placebo effect, so a reduction as a result of having the placebo treatment. But to control for kind of just relaxation across the study, other repeated measures effects, we had another group where we were totally truthful throughout. So they were randomized to receive a saltwater spray and were told that it would be saltwater. We told them that they were having air, so if they were less anxious, that's why. And then we told them that they were going to have the carbon dioxide again at the end. It was completely open throughout. And we thought we might see a difference between these groups to show that the placebo has worked. And what we found was surprising to me and not what we hypothesized. These were all our outcome measures from the CO2. Y-axis is anxiety or mood. The higher the number, the worse it is. X-axis is time. So this is before CO2, after the first CO2, after air, and after the second CO2. And what we basically found was that the repeatability of the CO2 model is really, really good. There was no difference between the groups whatsoever. In fact, I was very impressed with how little difference there is. Um, I think we did a post hoc power analysis, and we found out that to detect a difference between the groups, you would need 1,000 people per arm. So it's minuscule. Um, so that was not what we intended to find out, but it was kind of nice in its own way. Um, Interestingly, though, we looked at expectations and we measured expectations before they received the nasal spray and after conditioning. So we asked them, um, this was before the nasal spray and afterwards, now that you've had the nasal spray, how well do you think it's going to work in the final inhalation? And we saw in the group that had the lorazepam, we saw a significant increase in expectations. Um, so that looks like the conditioning worked. And obviously, the saline group, there was no difference in beliefs. I mean, these people who were kind of in the upper half, I'm not sure what they were thinking, but certainly the overall there was no kind of improvement in expectations there. So that sort of looks like the conditioning worked, but it didn't translate into a placebo effect for some reason. Um, we looked at kind of within participant changes, and there was quite a lot of variation. So um, this was a measure of CO2 reactivity, so change in the measure from before the inhalation to after. And we had to look at this change because there were some subtle differences at baseline between the first and second inhalation. But you can see some people really, anxiety really increased, whereas for other people it really reduced. And we didn't collect this routinely. And I was blinded throughout, but people who were helping me rate participants' anxiety, they told me afterwards when I was unblinded that some participants reported catastrophizing. They kind of said, "Oh, it feels like the treatment's not working in the second inhalation," and so that made their anxiety worse. So maybe there was a kind of interaction, or maybe as a result of a contrast effect. It was almost the air and the CO2 were too different from each other for it to be believable. Maybe. Um, the other thing I was thinking, and I started to think when I kind of thought about this more, was that maybe it's just not very easy to induce a placebo effect on anxiety itself, because we know that anxiety is associated with reduced placebo effects and increased nocebo effects. That's probably mediated by autonomic response. And as I showed you, inhaling CO2 increases blood pressure and heart rate. So, and actually getting anxious tends to increase blood pressure and heart rate. So, maybe actually just inducing a placebo effect on anxiety is just really difficult in a lab. But then I thought, well, but we see such big effects in clinical trials, so it must be possible. There must be something else. Um, and then it just so happened that we've recently finished analyzing a study where we were exploring transcranial direct current stimulation for social anxiety. So this is kind of a low-level neurostimulation aimed to help improve kind of emotional regulation. Um, And we did this, we gave this to people before they did a social anxiety challenge that I designed during my PhD. This is a picture of it here. I had a lot more hair at the beginning of my PhD. That's what a PhD does to you. Um, and the way this worked was we essentially asked people to get to know someone on Zoom, Microsoft Teams, while being watched by three other people. Um, in reality, those people are kind of dummy accounts logged in online without the participants' knowledge, but they're not actually being observed, but they're there to kind of induce that kind of feeling of social evaluation, and it makes people very very anxious doing it. And we wanted to see whether TDCS treatment beforehand can help improve the anxiety that people experience during the challenge itself. Um, and what we found was quite surprising. We didn't find any effects of TDCS, um, but we found a very nice effect of what you thought you had. Um, so this was again kind of y-axis is anxiety, x-axis is time. So this is after TDCS. We do an anticipatory period where we tell them they're about to have the interaction before they have it, and then there's the after the social interaction. And you can see across this was across all treatment arms. If people believed that they had the sham, they felt more anxious after the interaction compared with people who believed they had active TDCS, uh, which was quite nice. So we thought, oh, we've found a placebo effect in an anxiety challenge, which is good. Um, and we then kind of looked at what's the interaction with the actual treatment? Is there one? And it does appear that there is one. So this is again the anxiety after the interaction on the y-axis. Here is what people perceived they had. So these group of people perceived they had active. This group of people perceived they had sham. And then the colors are the actual assignment. And I think this is quite interesting. So we actually, if people had active TDCS, we found no difference in their anxiety, whether they perceived they had active or perceived they had sham. But there was a big difference in the sham group. So if they believed they had sham, they felt much worse, and that's significant. Um, there also seems to be like a ceiling effect, so that the active TDCS didn't seem to improve things more than more than believing that you'd had TDCS, um, which is potentially quite interesting and calls into question the additive model that we have maybe in TDCS. The caveat to that is these were all healthy volunteers, so maybe in patients it differs. But I think that's potentially something quite interesting, and we're looking to explore more about how beliefs might impact response to TDCS. Um, so just to summarize, I think overall, placebo effects in psychiatric disorders, there is much, much more to do. Um, we definitely need more data about expectations in clinical trials, about blinding integrity as well. But I think we also need more mechanistic studies in psychiatric constructs, so that we can really understand, what are the mechanisms driving these phenomena, because they might be useful as treatment targets as well. Um, so with that, I just need to thank everyone who's supported all these studies, my supervisors, David, Matt, Samuele, everyone who helped collect data, and my funders as well, the MRC, NIHR.